So the first operon that I'm going to talk about is the trip operon. So the trip operon basically produces tryptophan, and in its natural state it's on, which means that the RNA polymerase is bound to the promoter, and it allows the synthesis of tryptophan, which through the synthesis of mRNA, which makes the polypeptides that make the enzymes that make tryptophan. So the regulatory gene in this case does make an inactive repressor protein from mRNA, but as I said, it's in its inactive form because nothing is bound to it. So how you turn the operon off is the excess tryptophan acts as a co-repressor and binds to the regulatory protein, which is an allosteric protein, which means it has an active and inactive form, and by binding to it, it activates the repressor. And now the repressor is going to bind to the operator. And when the repressor binds to the operator, it stops the RNA polymerase from binding to the promoter, which halts the production of tryptophan. So this makes the trip operon a repressible operon because when the repressor protein binds to the operator, it directly inhibits the production of tryptophan. So this whole process is a negative regulatory gene expression because of the repressor binding to the operator and halting tryptophan production. So the second operon that I'm going to talk about is the lac operon. And the lac operon produces lactose. So in its natural state, the lac operon is off, which means that the regulatory gene has produced through using mRNA has produced the repressor protein, and this repressor protein is already in its active form to begin with. So the repressor protein is bound to the operator, and this prevents the RNA polymerase from binding to the promoter, which stops the production of lactose. So how this operon is turned on is basically allolactose or lactose acts as an inducer and binds to the active repressor. And when it binds to the active repressor, it causes this active repressor to come off of the operator. So it comes off, and it changes it because this is an allosteric repressor protein. So it changes it from its active form to its inactive form. So now it's in its inactive form. So now the RNA polymerase can bind to the promoter, and so now it can produce the synthesis of mRNA, and that can be used to make B-galactosidase, permease, and transacetylase. So what B-galactosidase does basically is it catalyzes the lactose into galactose and glucose, and the permease transports the lactose into cells, and the transacetylase breaks down the lactose by adding an acetyl group. So the lac operon is an inducible operon because the lactose that binds to the repressor is an inducer, because when it binds to the repressor, it causes the repressor to leave the operator, which allows the RNA polymerase to bind to the promoter and allows the synthesis of lactose. And this whole operon is still negative gene regulation, because when the repressor protein in its active form is bound to the operator, it directly stops the production of lactose. The operon that I'm going to talk about is the CRIP operon, or the cyclic AMP receptor protein operon. So basically what happens is when glucose levels are low, CAMP or cyclic AMP accumulates, and when it does, it acts as an activator, and it binds to CRP or cyclic AMP receptor protein in its inactive form. And when it does this is an allosteric protein. So when the CAMP binds to it, it changes its form and becomes active. And in its active form, it binds to a site upstream of the lac promoter. And by its binding, it actually causes the DNA to bend, which makes it easier somehow for the RNA polymerase to bind to the promoter. And this directly stimulates transcription of the lac operon. Okay, so then what happens though is when glucose levels rise, the concentration of CAMP decreases, and this causes the crypt in its active form to become inactivated because the cyclic AMP is gone, and then it disattaches from the promoter of the operon, and this causes the RNA polymerase to also disattach from the operon, and this causes the transcription rate to go down. So it's basically like a volume control, and it turns the level at which transcription occurs, it turns it down. And this is positive gene regulation because when the cyclic AMP receptor protein binds to the promoter, it directly stimulates the transcription. So this is positive. This is an example of positive gene regulation. So the process of RNA splicing is basically how pre-mRNA is modified. So pre-mRNA, big parts of it are cut out from the original RNA that is synthesized, and this process is called RNA splicing. So in the pre-mRNA, there are many non-coding regions called introns, which are interspersed between coding regions called exons. So in the process of RNA splicing, these introns are cut out of the sequence, and the exons are spliced together to make the mature mRNA. So how pre-mRNA is modified? So basically what happens is the pre-mRNA at the 5' end, a 5' cap is put on that end, and that's a modified form of a guanine nucleotide. And this 5' cap serves two purposes. So one, it protects and prevents the mRNA from degradation, and it also acts as a binding site for ribosomes. And then what's happening at the 3' end? So basically at the 3' end, an enzyme adds a poly A tail to that end, and the poly A tail also prevents degradation of the mRNA, and it also acts as a way that the pre-mRNA can be exported from the nucleus. Okay, so basically what happens is that RNA splicing is when a large portion of the primary transcript or pre-mRNA is cut out to make the mature RNA. So for eukaryotic organisms, they have many non-coding regions called introns interspersed with coding regions called exons. So the exons, so RNA polymerase basically transcribes the coding and non-coding regions from the DNA template, but the introns are cut out and the exons are spliced together. So this is the process. So at the end, so the signals for RNA splicing are located at the end of the introns in short nucleotide sequences, and small nuclear ribonuclear proteins recognize these splice sites. And the small nuclear ribonuclear proteins are called SNRPs, and they are comprised of protein and RNA. So basically what happens is the SNRPs bind with other proteins to form a larger complex called a spliceosome. And what the spliceosome does is it interacts with the splice sites at the end of the introns, and it cuts the introns at these specific sites, and it cuts them out. So this is the excised intron, and it splices together the exons to make the mature mRNA. But one polypeptide is not always the result from RNA splicing due to an occurrence called alternative RNA splicing, and that has to do with which segments are considered as exons and which are considered as introns. Because if separate segments are considered as exons, that would change the segments that are spliced together, thus changing the resulting polypeptide.